so hello everyone, and welcome to this webinar session. So today we will speak about automated fluid delivery for your cell and tissue imaging. Okay, so my name is Mélanie Chabot. I am an application engineer at Fluigent, and I will be assisted today by William Cesar, R&D project manager, and Noé Viovi, R&D software developer at Fluigent. So our program today will be as followed. First, I will tell you about the reason and advantages to use automated sequential fluid delivery for your cell and tissue imaging experiment. I will then show you two demo videos of our solution to achieve such objectives. And in a third part, I will mention three examples of application in cell biology, such as multiplex tissue imaging. We will then have around 20 minutes left to answer your question with William and Noé. Okay, so let's speak about the reasons and advantages to use automated sequential fluid delivery. As you probably know and have directly experienced, the typical immunostaining workflow, which allow you to capture those beautiful images, such as the mouse testis section here or the whole mouse embryo here, is a workflow that necessitates a succession of multiple pipetting steps. And with manual pipetting comes huge disadvantages. First of all, the injection of fluids is not controlled, not properly balanced, and you can create very high flow rate when you push onto the plunger button. So indeed, you can manually push up to 1 ml of liquid in few seconds, and this creates a very high shear stress on your samples. In addition, the injection is rather uneven, which increases the variability both in between experiments that you will perform when the same person does the experiment, and even worse when comparing experiments performed by different operators. And the flow is that is created by manual injection is turbulent, so and this can damage your sample. Another issue with the pipetting comes from the geometry of the tips. The conic shape results in a non-homogeneous velocity distribution, and therefore high shear stress again is present at the tip hole and on the sample. And in addition, there are always risks of pipetting errors, solution mismatch, and potential contamination. There is also issue with liquid injection while performing your live cell experiment. It can introduce some bias. Indeed, the pipette tip can displace the dish and affect the original imaging position. Sample can be flushed away. You can have liquid that spill over the microscope or on the objective that are just located below the sample. And in addition, there is always a risk of contamination when you open the lid of a plate or a dish to inject the liquid. So to overcome this issue, Fluigent has created ARIA. So I will play this quick commercial video to introduce the system.
Introducing ARIA, our perfusion automation tool. ARIA makes it possible to automate delivery of up to 10 different solutions in a petri dish or microfluidic chip by following user-defined protocols. ARIA's precision makes it possible to inject defined volumes at specific times and to provide time-based sequential perfusion of multiple fluids under controlled flow conditions. ARIA's intelligent software allows you to design the protocol that best fits your experimental model. Reducing manual sample handling and procedural complexity is key to improving precision, increasing reliability, and saving time. Even the cleaning is automated. ARIA perfuses successive pulseless flows of different solutions, avoiding the extensive mechanical stress associated with classical pipetting. ARIA automates protocols such as immunostaining by programming the time delivery of primary and secondary antibodies, the injection volume and flow rate, as well as the incubation time.
All right. So yeah, as mentioned in the video, ARIA can really be used as an automation tool for immunostaining. It offers a high precision since you can get a complete control over the fluid injection parameters. You can choose your flow rate, the volume and the timing of injection. It's also a time saver since you can fully automate the protocol. It's extremely user-friendly. The hardware and the software are very intuitive. They have been designed for people that have no expertise in microfluidics and doesn't require any specific training. It's quite versatile since you can inject up to 10 different solutions. You can use our standard protocol, but the aim is really to design your own custom protocol. And finally, but very importantly, ARIA provides the reproducibility and the reliability that are so important for precious experiments, especially when you have precious samples, donors, mice, and you don't want to miss an experiment in that case. So I will show you how easy to design an automated program for your immunofluorescence protocol. So take the classical immunofluorescence workflow as you would write it in your lab notebook. And so here you just generate your protocol by adding sequential orders. So it's an illustration here where you will start with injecting PFA, for example, for the fixation step. You define the flow rate and the total volume. Then you can have a wait time, which corresponds to the incubation time. So for example, 30 minutes, which is classical. Then you do an injection of PBS to wash for 10 minutes, for example. So it's really simple. You just add the orders sequentially and then inject antibody 1. You define the flow rate and the volume, and again, add an order of wait for an hour for the incubation. And again, for the secondary antibody. So yeah, it's really, really simple and straightforward. So with ARIA, you can work with a minimal volume of 40 microliters. The flow rate, ranges from 3.2 microliter per minute to 80 microliter per minute with the M version, or to 40 microliter per minute to 1 ml per minute with the L version. It's possible, as I said, to inject up to 10 different solutions, and you have both a manual or a software control. You can also inject just manually the solution by pressing on the right button. But again, the aim is to automate the protocol, so to use the software that comes with it. We have 2 versions that are available. The single output version that is perfect for high quality multiplexed image experiment, or the serial output version that is perfect for multi-sample perfusion, since you can connect up to 9 samples. We are also open to collaborations, and if you want, we can also work on a customized integrated system based on your specific needs. So this we can discuss in more detail about that if you're interested. Okay, so now I will play a demo video which describes really ARIA and software. So I hope it doesn't play too fast. I might do it manually, actually. Yeah, I prefer to do it manually. Okay, so here is the ARIA software that I told you is very intuitive. And so you can create your automated protocol. You simply add the fluid delivery orders, incubation time, TTL signals, And so to illustrate the procedure, so once you have set everything and the calibration is done, you can start to inject the delivery program. So here you see with the dye, the liquid that is going through the fluidic line into the chip and coming out. And so you can inject sequentially several solutions. So here it's just dyed again to illustrate the procedure. But everything is really automated. You can also choose to flush your tubing in between 2 solutions to avoid mixing between the 2 solutions. And here I just wanted to illustrate the sequential delivery of different solutions in a flow chamber. that was kindly provided by Bioptex, a partner. So you can see, again, the sequential delivery of the solution that are flowing along the chamber as you would do for your antibody solution. So it's to perform immunolabeling. Okay. So about the application. So we'll briefly introduce 3 different applications where ARIA acts as a beneficial tool. The first application is an example of spatial proteomics. It comes from a recent method paper that was published by the lab of Ron Germain at NIH in a Nature protocol in February 2022, and that was further highlighted in a special issue of Nature Methods in March 2022, and it relies on the automated multiplex tissue imaging. So first of all, what is spatial proteomics? So classically, on one hand, the cell and tissue imaging allows you to visualize in situ proteins, but can only reveal the localization of few proteins. And on the other hand, there is proteomics that enable to analyze multiple protein expression, but not in their native form within the tissue, as it necessitates protein dissociation. So in order to overcome these issues, several new protocols have been implemented to perform spatial proteomics to visualize multiple proteins directly in situ. So the aim of spatialomics is to localize multiple proteins within tissue, which is not possible with classical immunofluorescence that is limited to 4 or 5 colors. So the question is how to scale up. So today the example is called IBEX for iterative bleaching extends multiplexity, where it's actually a succession of multiple cycles of immunolabeling, image acquisition, and fluorophore inactivation, bleaching. So this is done manually, and IBEX can enable to multiplex, to have a multiplex imaging of more than 65 parameters. But in the paper, they also have automated the sequence by coupling ARIA to a microscope. And so it's, again, the succession of multiple cycles of immunolabeling, image acquisition, and fluorophore bleaching. And so ARIA communicates with the microscope through TTL signal that is sent and received to launch the acquisition and to resume the perfusion sequence. So, sorry. So what you need is to interface a fluidic device with a microscope. So you create your protocol using the ARIA software, as I showed you, and that will then control the automated perfusion system to sequentially deliver fluids into a flow chamber or a microfluidic chip that is placed onto the fluorescent microscope. And this will interact with our perfusion system. So here is the setup that has been used by Radke et al., where they have mounted their tissue onto a specific flow chamber, place it under the microscope, and use ARIA to deliver sequentially the liquid. So here is the protocol, the ARIA protocol that they have used. So first they inject PBS to rehydrate the tissue. Then they perform a nuclear staining and using UXT that is injected at a certain flow rate. They wait 5 minutes, incubation time of 5 minutes. Then they wash with PBS. And yeah, they washed with PBS. They did a wait for user step just to define the different region of interest on the microscope software, and to prepare the Z slices, to choose the Z stack. Once this is done, they resume the perfusion and inject their first antibody. They wait for an hour to incubate it. Wash with PBS, and directly, automatically, at the end of the PBS wash, the imaging procedure will be launched, since they have activated, here you can see the bell that is ringing, to launch the TTL signal. Once the microscope has acquired the different Z stack in the different position, that have been predefined. You see here the wait for TTL order. So it will it will give the instruction to ARIA to resume the injection, the fluid injection protocol. And again, so restart with the PBS wash, injection of the bleaching agent. So it's lithium borohydrate. And a PBS wash, and again antibody 2, and the cycles are repeated again and again for all the different antibodies. So normally, once you have set the ARIA protocol, you press start, and everything should go well, and your images are taken automatically, and the immunostaining is performed automatically. So these are the example of images that have been obtained using this approach. We can see here the different zone and cell compartment of a human lymph node. Here we see the structure and the localization of protein that forms the small intestine or human skin. And so they were able to image more than 20 proteins successfully and automatically here. So to conclude, the automation power of ARIA here clearly benefits their protocol. They have compared the manual approach and the automated approach, and it's a gain of time of 2 hours per cycle, so it's quite a lot. And also it provides high reproducibility by delivering the fluid in a steady and sequential manner, which minimize the pipetting errors and even the solution mismatch. Okay, so another application, another example of spatial omics is spatial transcriptomics, and more specifically seqFISH. So the goal here is to determine in space where are each RNA of interest within a cell or a tissue. And to do this, there are 2 main approaches in spatial transcriptomics. A multiplexing method, such as seqFISH and MERFISH, that relies on a barcoding method that allows the multiplexing. So the major advantage of this method is that it limits the number of cycles to be performed in order to extend the number of RNA species to image. It is a genome-wide screen, as it is possible to look at around more than 10,000 transcripts. However, it requires a lot of unique primary sequences. Then there are the amplification methods, such as StarMap and cyberFISH. And here, probes directly target RNA and amplify the signal. A major drawback of this approach is the limited number of detectable genes, but it mainly depends on the application. If you don't need to have a genome-wide screen, then the amplification method is enough. So seqFISH belongs to the multiplexing method, as I mentioned, and it relies on temporal barcoded RNA probes that undergo multiple rounds of hybridization, which allow many proteins to be multiplexed. And the main benefit of seqFISH is that it scales to the genome level, as I said, up to 10,000 genes. However, it necessitates high magnification and numerical aperture to resolve all the fluorescent dots. It's also quite a long experiment that it requires more than 80 cycles, 80 rounds, so it can take several days. And so here is the setup of our collaborator at EMBOL Rome, of Alvaro Crevena, who has used ARIA combined with Bioptex chamber with a Nikon Ti-E microscope, and in this configuration, so to perform his seqFISH. And here in this configuration, the main limitation is actually the manual change of probes after a few cycles, because as I said, it requires 80 rounds of probe attaching to the sample. So yeah, after a few rounds, you would need to replace manually the probe reservoir. But still, it's a gain of time. It's still a gain of time as compared to manually. So if you wish to know more and better about spatialomics, I suggest that you watch Alvaro's webinar that is available on our website. So here is he's really the expert, and you will learn a lot. Okay, and a last application, really different, that we have developed in collaboration with the team of Stéphanie Descroix at the Institut Curie is the capture and characterization of circulating tumor cells. So the context, in aggressive metastatic cancer cells from the primary tumor can travel through the blood circulation and form new secondary tumor on other parts of the body. And the detection of those CTCs for circulating tumor cells is an indicator of a potential metastatic status of a cancer. The number of CTC is really important. It defines a threshold under which the probability of survival is high. So this is set to 5 CTC in 7.5 ml of blood, quite precise. And above this threshold, the probability of survival drastically decreases. It's also important to decide which therapeutic strategy will be chosen. So for patients that have less than 5 CTC, an hormonal therapy will be proposed, whereas for patients that have more than 5 CTCs, chemotherapy will be administered. And so the main challenge in detecting and counting these CTCs is really their low proportion among the total blood cells. So for example, if in 1 ml of blood, there is approximately 1 to 100 CTC compared to the 5 billion of red blood cells, 0.2 billion of platelet cells, and 7 million of leukocytes. So CTC are really rare biomarkers, and microfluidics is a powerful tool to detect a rare event like that due to the high sensitivity. So the team of Stéphanie Descroix has developed a chip to capture and characterize the CTC. The chip is made of 4 chambers, and each chamber possesses a network of magnetic beads. So if we look at the transversal cut, we can see the column of magnetic beads that are coated with anti-EpCAM antibodies, because CTCs express a lot of EpCAM at their cell surface. And so when the blood when the patient blood flows inside the chip, the CTC will be trapped by the beads that are coated with the antibody. And so once the CTC have been trapped, they can either be labeled or analyzed by DNA hybridization directly within the chip, or even cells can be retrieved for further analysis. So this is a multi-step protocol that is based on sequential fluid injection that can be automated with ARIA, and that have been automated with ARIA. If you want to know more about this chip and this approach, I suggest you read their paper. Nice. So the different steps that have been automated are the surface treatment of the chip and the incubation, then the injection of the magnetic beads, injection of cells, the fixation with PFA, and the whole immunostaining procedures. So it's a real time saver. They save 2 hours in the process here. So here are the images. And so you can see that the cancer cells were trapped on the magnetic beads and successfully immunolabeled. We see actin in orange, the nucleus in blue, and the damaged DNA in green. And so if you want to know more about this application, you can check the application note that is available on our website. There are several applications that could benefit from the automation capacity of ARIA, such as shear stress experiment. Since you can control the flow rate, you can control also the shear stress that you impose onto your cells. You can image cells for a long period of time. You can use it as a perfusion system, especially if you have a complex 3D cell culture. It can also be used to automate pulse chase experiments for live cells. You inject a drug, you image the cell reaction, and perform washout and see how the cell resume its motility, for example. It can also be used for DNA paint, for super resolution. The application, there are no limits in the application, actually. It's just a matter of setting, okay, so to conclude this webinar, the take-home message is that ARIA can help you save time. You can create automated protocols. It reduces errors, improve the precision, and this make value each of your experiments. You can also share easily protocols with a partner worldwide. And so if you have any questions about the system, or if you want to wish to obtain a quotation, just send us an email at contact@fluigent.com. And if you wish to discuss more about the applications, or if you have a collaborative work to propose us, please contact me directly and at melanie.chabot@fluigent.com, and I will be happy to discuss with you. Okay, so I would like to thank you for your attention, and we will be pleased now to answer your question. So please ask them through the Q&A panel, or raise your hand, and I will give you the lead. Thank you. So I see that there is a question. Sorry, so you may provide it, but it is not obvious from the pictures. Can you control the temperature of the solution stored on the machine before injection? So this is a good question. I don't know if William wants to answer. I think I can answer.
Yes, I can answer. So hello, everybody. So the machine itself right now cannot heat the solution and keep it heated before injection. So there is no temperature control associated with it. We are currently thinking of an accessory that we will be able to adapt to the reservoir so that this becomes possible, but right now it is not possible. What you can do, maybe if you want your solution to be really heated when it enters the chip, is to have your chip inside the incubator and have the tubing long enough so that it gives time for it to be heated before reaching the chip. But right now, no solution for heating really the input solution.
But we are developing it, or we are thinking of developing it. What about CO2 for imaging live? William?
Well, about the application, I don't know it. I cannot really.
Yeah, well, I can maybe answer.
If it is about mixing CO2 inside the solution, it is possible, as ARIA has a pressure input where you basically have to inject gas, and CO2 is compatible. So it is possible to put pressure with a mixture of air and CO2, for example, and to have your solution that is like you have some dissolved CO2 inside of it, but it's not precisely controlled. You just passively inject the CO2 solution mixture, I mean, in the solution. I don't know if I am really clear, but
yeah,
the application, I don't know it personally.
Yeah, otherwise the chip, I mean, if you connect ARIA to either a flow chamber or a microfluidic chip, you place it under the microscope, and you have the top, the classical CO2 top that you can add. Normally, we have people, how to say, constructors such as Ocolabs or Pecon they provide a specific old insert that have space for the tubing, so that allows tubing to be inserted into their chamber. And I used to do that when I was in a research lab, and this is quite convenient. And so you have CO2 as you would do on your dish. Okay, so the next question. For antibody solution injection in IBEX method, first example, a lot of antibody solution needs to be prepared to fill the tube, which is a waste of antibody. Is there a way to reduce such waste? To be prepared to fill the tube. So it's not that much actually. The minimal, you will correct me if I'm wrong, William, but the minimum volume is 40 microliter.
Yeah, so the minimum volume to inject in the chip to be able to inject a precise volume is 40 microliter, but the internal volume of the machine itself is at least 100 microliter. So then it depends on the length and the volume of, the internal diameter of the tubing you connect after the machine, because you can have a very long tubing if you wish, but it will, of course, add some more volume to reach the chip. So I would say you anyway have to use at least 100 microliter. Yeah. So yeah, this is really the minimum you can work with.
Okay. I don't know if it's a lot, if it's too much for you. Yeah, we can also, it's true that we can also just, you can just push, you can inject your 40 microliter in the system and push with PBS. If it's really important for you to minimize the volume of antibody, it cannot be less than 40 microliter. This is due to the pressure, to the reservoir, and the fact that you put pressure in the reservoir to inject the liquid. But it's true that your solution can be pushed by the PBS, by the volume of PBS. I don't know if you have other questions. It can be about the software, or you can still contact us afterwards. Elisa, yes. Hi, Elisa, if you want to ask your question.
Hi, good morning. First of all, it was very interesting. Thank you so much for this presentation. So I work with microfluidics and the closed loop control of cell behavior. So I was wondering, I have two questions actually. The first one is how easy, and if it's possible at all, to interface your software with MATLAB or LabVIEW with something that you can customize even more the delivery of drugs. And second, if you actually, from one video, it was pretty clear to me that you can mix chemicals before reaching the cell culture chamber, eventually for live imaging. Is that true?
Yes. So for the first question, we'll give you about the SDK and how, it's mostly about SDK, but how you can interface with MATLAB or LabVIEW or Python, whatever.
Yes. So as I said earlier, for now we have no interactions through the SDK so MATLAB and LabVIEW are not yet supported and maybe should not be supported because it would disturb the automation process. If live control is required, or maybe a very specific application, maybe independent Fluigent components used with the SDK or the software Oxygen would be a better solution, because they support interactions with LabVIEW, MATLAB, Python, etc. Hope this answers.
What I what I can propose also, so I can directly discuss with you by email, Elisa, and we can see, because we have people asking this kind of question, and the thing is to develop more this part, we need to have a specific idea of the application behind to generate something useful, because people are asking for SDK, but we don't know for exactly in which context and for which specific application. So I think it would be good if we can discuss together to define more specifically your need, and this will help the development for the software team, because these are questions that are coming.
Yeah, that would be great. Thank you.
Yeah. And for your second question about the mixing, yes, indeed, you can clearly see, especially with the dyes, it's very easy to see that the solution are mixing along the fluid lane. And that's why, so this is real. That's why we suggest to do a flush. There is this order called flush tubing or tubing flush in between two solutions to be sure that they are not mixed. Because people have suggested to inject airs or oil bubble, but this is not compatible with the system. It disturbs the flow meter, the flow unit, and then the whole fluid delivery orders are messed up because the flow rate is not good anymore. So the best option for now is to include this flushing step in between two solutions.
Thank you. Thank you so much.
Again, if you have question that comes later, or you want to discuss specifically your application, your need, or specific features, please do not hesitate to contact us. We will be pleased to answer you and to help you. So thank you very much, everyone, for your participation, and have a good end of the day. Bye.